DNA. It's what makes us, well, us. It contains all the instructions our molecular machinery needs to build the complicated structures of the human body. But what if we have chunks of our DNA inserted or deleted? Our machines don't know how to build our essential proteins. So, what can we do to fix this? What does herpes have to do with it? And how are we using this to treat conditions like dystrophic epidermolysis bullosa? Welcome to the genetic basis of stuff and things. To start this video, I'd like to introduce you to a condition called dystrophic epidermolysis bullosa, or DEB for short. To understand how DEB works, we first must understand the function of the COL7A1 gene. COL7A1 is a gene of about 9 kilobases in length, corresponding to 2,944 amino acids. A normally functioning COL7A1 gene produces the protein collagen 7, which makes triple helical homotrimers, forming the anchoring fibrils. These structures help hold the dermis and epidermis together. What about those with DEB? Well, those with recessive DEB have two non-functional copies of COL7A1, mostly nonsense or phase shift mutations. Without a functional gene, they cannot make collagen 7, so they lack the anchoring fibrils. Their skin has a harder time healing, is more fragile, and more frequently blisters. Recessive DEB has a prevalence of about one baby born every 2 or 3 days. Now, let's talk about gene therapy, which is the technique of using nucleic acids to help treat medical disorders. First performed on humans in 1990 to treat a 4-year-old girl born with a severe immunodeficiency disorder, this field has grown massively. I'll be focusing on gene replacement therapy, which is all about fixing or replacing a faulty gene. But how do we get this replacement inside someone? Luckily, evolution has already solved this problem. When a virus infects you, it enters your cellular membrane before uncoating and releasing its genetic material. When the time is right for the virus to reproduce, its genes will take over the host machinery, producing copies of the virus, which will eventually assemble and lyse the cell, spreading to future hosts. Okay, this sounds bad. How does this help us? Well, the virus was able to bring the genetic material it was carrying into its host cell. What if we change the load that the virus was carrying? Scientists take the virus's genome, remove the genes that make it virulent and dangerous, and add the genes that they want in the host cell. In this form, the virus is called a viral vector, and there are many different vector types. Let's talk about a groundbreaking treatment for this called Beremagene Geperpavec, or B-VEC for short, which was approved for use in the United States in May of 2023. In this treatment, a modified form of herpes-simplex virus type 1, or HSV1, yes, the herpes you're thinking of, was engineered to be replication deficient. 2 copies of the complementary COL7A1 DNA were inserted into it. Following 2 trials with small sample sizes, a phase 3 trial was conducted where 31 DEB patients were sorted into pairs with similar wounds, and one of the pair received 26 weekly topical gel treatments containing the vector, while the other person received a placebo. So, theoretically, the vector is able to make its way into the skin cells of the patient. Now the COL7A1 complementary DNA is in the cell to be transcribed and translated. Due to how this particular virus works, the DNA is not integrated into the cell's genome. But you may be thinking, why are we using herpes instead of a different virus? Our body can't differentiate between the good viruses used for gene therapy treatment and the bad viruses trying to infect us. So, to be safe, it attacks them all. This has an array of consequences for the gene therapy. The body might attack the vector, triggering severe immune responses. Additionally, like a vaccine, the immune system learns about the vector when affected, allowing it to quickly identify and defeat it in the future. This makes giving repeat treatments particularly challenging, since with each successive application, the body may get better at fighting it off. But what about herpes? What makes it so different? Well, HSV1 is highly competent at evading the immune system, making it perfect as a vector. Even if the body has already encountered HSV1, The virus is so good at evading the immune system that it will still be able to infect the host without causing alarm, meaning that the treatment is redosable. So, with all this in mind, did the treatment work? Did treated patients receive a functional gene and start producing collagen 7, repairing their skin? Yes! After 6 months, 67% of primary wounds exposed to B-VEC healed completely, while only 27% of untreated wounds did. Additionally, those treated with B-VEC reported a statistically significant lower pain severity when wounds were redressed. This treatment marks a huge step forward in gene therapy. B-VEC is the first approved dermatological gene therapy. It is also the first approved redosable gene therapy, and the only DEB gene therapy working in vivo. The world of gene therapy is full of untapped potential, and we're only now starting to see the bigger picture. But we have a lot more work and discovery to do.